Hello, so today we're going to move on to gene expression. In your previous lesson, you learned about nitrogenous bases and how they pair, of course, where they are located in DNA. And today we're going to start talking about genes. So what is gene expression? Well, DNA, we know, controls all the heredity characteristics of an organism because each gene represents the code, or we could say the recipe for a protein. So the gene is going to be expressed when it directs the synthesis of protein in a cell. So gene expression is really protein synthesis. And remember, synthesis means to make. So a gene is a section on DNA that codes for a particular protein, basically meaning it holds the recipe. So gene expression is the way that a gene becomes the actual trait. So what we're actually seeing in our friends and our family. It is what happens when the gene becomes activated, and that just means turned on. So when genes become activated, they cause proteins to be made, and these proteins are the actual genetic traits. So DNA, which is made up of genes, make proteins that make up traits, which essentially makes up each individual characteristic that humans and other organisms have. Please remember, it's very important that synthesis means to make. So here's an example. Eye color. So DNA codes for the color of your eyes. Color is based on a pigment, okay? So it's light pigments or dark pigments that are inside the eyes. In order to have that pigment in your eyes, your DNA must have the recipe, so must have the code for the proteins that make up the pigment, which gives them their color. So DNA must have the message or the code or the recipe to carry a specific eye color pigment. If it carries this specific eye color pigment, like blue, green, brown, that means it has the proteins to make the pigment. So what do we remember about proteins from our macromolecules unit? Remember, proteins are very specific. Proteins consist of chains of amino acids. There are 20 different amino acids. And each amino acid is slightly different because of their R group. What we have circled on the bottom of the screen in pink is the different R groups for each of the amino acids shown. So amino acids are held together by chemical bonds to make polypeptides. If the polypeptide is at least 100 amino acids long, it's called a protein. So remember, amino acids are the monomers that make up long chains called polypeptides. And once those amino acids reach at least 100 amino acids in the polypeptide chain, then we will call it a protein, which is a large or a macromolecule. So when amino acids bond together, this is through a process of synthesis, specifically dehydration synthesis. We're removing water molecules from the amino acids to form a peptide bond. So when you eat protein like chicken, remember it's digested into these small monomers, which are the amino acids. So synthesis is to make, digestion we're breaking. The sequence or the order of amino acids in a protein determines its shape. The shape of a protein determines its function. The function is also the job. So the sequence that the amino acids are within a gene determines what the shape of the protein is. Therefore, will determine what the job is inside of the cell for that protein. So here's an example of the shape determining the function. So the first target cell, we see our hormone A attaching to the receptor for hormone A. For a target cell that targets A and B, we see hormones A and B attaching to the receptor site. For target cell B only, hormone B only, we only see hormone B attaching to the receptors because remember, The different order of amino acids determine the function because of the shape. So order of amino acids determine the shape and therefore determine the job of the protein. So we also have structural proteins in the cell membrane. And again, they have different functions because they're carrying out different jobs because of their different shapes. Enzymes are another type of protein, and they are going to act as catalysts, and catalysts are like a puzzle piece. So enzymes fit to specific substrates, and remember, enzymes help speed up reactions. Hormones are another example. We have estrogen, progesterone, and testosterone. Pigments, such as hemoglobin, which we can find in red blood cells. Eye color and skin color are also made up of pigments. So how does protein synthesis work? Well, the DNA code, which is the recipe to make the protein, is needed in order to make these proteins, right? So the DNA is located inside of the nucleus. Remember, proteins are made in the cytoplasm. So we have DNA in the nucleus, protein is made in the cytoplasm at the ribosome. So DNA directs the cell to make protein, but it's stuck in the nucleus. It can't leave the nucleus. So the proteins that are outside, that are made outside of the nucleus, in the cytoplasm at the ribosome site, need to communicate. So DNA inside of the nucleus has to get out, information has to get out to the ribosomes to make these proteins. So since DNA cannot leave the nucleus, the code for a particular gene is read by a special enzyme, and it's used to make a messenger. This messenger is known as mRNA or messenger RNA. Messenger RNA can leave the nucleus and deliver the code or the recipe to the ribosome where proteins are synthesized. So messenger RNA attaches to the ribosome, and a helper RNA, which is known as transfer RNA or tRNA, deliver the amino acids to the mRNA according to the secret code. Every 3 bases of mRNA is called a codon, and it is the code word for one amino acid. So at the bottom you see the codons, AUG, that's one codon. That codes for one specific amino acid. ACG codes for another specific amino acid, and so on. Now, please remember the 3 types of RNA. We have mRNA, rRNA, and tRNA. mRNA is the messenger, rRNA makes up the ribosome, and tRNA is the helper. So what molecule contains the recipe or instructions for making protein? That's known as DNA. It's inside of the nucleus. What molecule carries the instructions out of the nucleus? That's mRNA, messenger RNA. It can leave the nucleus with the secret message. Where are proteins made? At the ribosome. What are proteins made of? Amino acids. So remember, amino acids make up a polypeptide chain. Once that chain reaches 100 amino acids or more, it has to be at least 100, then it will be considered a protein. What molecule delivers amino acids to the ribosome? tRNA or transfer RNA. And finally, we're going to look at the amino acids and codons on mRNA. So this is the universal genetic code chart. We use this for mRNA only. You'll notice there are no T amino acids or thymines because we are looking at mRNA, and RNA does not carry T or thymine. So you'll notice each of these codes, so we have our first base, our second base, and our third base. Each of these bases make up a codon. This codon stands for specific amino acids.